So very good morning, Jen Dobry, to everyone here in the room, and a very good morning, afternoon, or evening if you're online, and depending where in the globe you are. Welcome to the session, because now it's time to dig into One Health. And we know that One Health is this, or we want One Health to be this transformative approach where collaboration, communication, capacity, and coordination is done differently. But for that, we need to bring all the disciplines and all the silos on board. One Health is about connecting this. And today we will therefore go into the one that some sometimes call the forgotten silo, the orphan, the lost child, the one who shall not be named. But the environment really is key. And there is more than enough evidence, as we know, to understand the unique impact that ecosystems and the environment has on the lives and on the health of all living creatures on this planet. And I'm very happy that we have an amazing set of colleagues today that will take us a little further into showing this connectivity, also highlighting what, of course, is one of the most relevant issues on environment today, which is climate change, which, as we know, at least on this side of the Atlantic, it's a reality. And it's something we should all be worried about. To help me with this interesting discussion, I'm very happy to have my co-chair, Taira Krause, that I think most in Eskaide will know. She's a famous face in Eskaide, a professor here in Copenhagen University. She's a researcher at the Serum Institutes and also the representative of Denmark in the advisory forum of ECDC. So very much happy to have you with me, Taira.
Thank you so much. And then I'll be very happy to introduce the speakers that we are going to hear today. So we'll start with a speech from Jan Semenza from Umeå University that will be on the nexus of climate change and One Health. And then we'll have a talk by Gerardo Sanchez Martinez, who'll give the perspectives of the European Environment Agency on the One Health approach. Then we'll have a talk by Frank Møller Aastrup, a professor at the Danish Technical University, who's going to talk about the potential of AMR surveillance using wastewater. And finally, we'll have Budwin Katri, who's going to talk about the Belgian One Health AMR surveillance program. So, and during the sessions, feel free to ask questions using the app, and it's also possible to use the microphones if you have technical issues with the app.
Thank you very much. Let's not make the audience wait much further. Climate change, Jan, for the next 25 minutes, it's all yours.
Thank you, thank you. It's great to be here. I'm the orphan here on stage. Thanks for inviting me. It's great to be here. I'd like to thank the organizing committee to invite me, specifically the chief scientist who sits here in the front row, Piotr Kramatz, my old friend. Thank you for the invite. It's great to be here. So today, I would like to speak about the nexus of One Health and climate change. And I've been asked to bring up this Venn diagram of One Health right up front, so we get this out of the way. Here it is. So now we are done. We have all seen it, right? So this is the concept, right? As we just discussed earlier, we need to make sure that we integrate human health with animal health and environmental health, because we can't work in silos. We need to cross these disciplinary divides and bring these different disciplines together, because they're interconnected and interdependent. So we need to make sure that human health interacts with veterinary services and environmental health services in order to make those connections, because if we do, we will be able to track epidemic precursors of disease and able to build early warning systems that can tell us where the threats are coming from, so we can act proactively and stop these outbreaks from happening in the first place. And the key, the Achilles heel of this concept is that we need to be able to integrate human health data with animal health data and environmental health data. That's the Achilles heel. we need a system, an interoperational system where these data systems speak with each other. And so we need to connect the human data with the animal data, with the environmental data in order to make sense out of them. And I haven't seen anyone build a system like this. This is something I've been advocating for decades, and we haven't achieved it. So I'm sorry to break the bad news for you guys. This is not what I will be presenting today. I will try to make a simple step how we can integrate environmental and animal data, entomological data, into these epidemiological assessments to get a handle on the risk. And based on this Venn diagram of One Health, we now merge this or embed this into the IPCC framework for climate change risk. Here it is. So in this diagram here now, we have the backdrop of the IPCC, the Intergovernmental Panel on Climate Change, risk framework, where we look at climate hazards, which is extreme weather events, temperature, precipitation events, and so on, that interact with vulnerabilities in societies, individual level vulnerabilities, infrastructural vulnerabilities, the predisposition to be exposed to risks. And so those vulnerabilities need to be taken into account besides the exposure to be adversely affected by those hazards, because only then we can assess the risk at the heart of this Venn diagram by taking into account human health, animal health, and environmental health. This is a framework. And I have to admit, I'm Swiss, so I can't get around the temptation to present the Swiss cheese model. Here it is. So here, this is the same concept. And here you see the cheese slices, right? At the very top, you have the climate hazards, again, it's climate extremes. Then you have the exposure level, the vulnerability level, and the risk level. It turns out that those exposure and vulnerability levels, they pose potential barriers to those climate risks, these climate hazards. Because guess what? If those holes and these pockets in the exposure and vulnerability horizontal levels don't line up, they will obstruct the impact of these environmental hazards. And unless those environmental climatic hazards find an opening in those gaps, unless that hazard finds an opening by time and by space, that's the thing that determines the risk that eventually puts population at risk. So this is the diagram, the Swiss cheese model, that basically reflects that framework that we're working with. And I would like to give you some examples, but I will always come back to these different layers of exposure and vulnerability throughout my talk. And the good news is that there are certain things we can do about these elements of the IPCC framework. The hazards can be mitigated through climate change mitigation, which means reductions of greenhouse gas emissions. For the exposure and vulnerability, we can apply public health adaptation and preparedness measure to reduce exposure and vulnerability, and then obviously risk management when it comes to the management of that risk. So on this slide now, at the very top, you have the climate hazards lined up. You can see here severe weather, extreme heat, rising sea level, environmental degradation. These are the hazards. And they predispose populations to specific health endpoints. You can see here heat-related mortality and a whole slew of different endpoints. I obviously can't go through them all today. They only gave me 25 minutes to speak today. So I will just focusing on changes in vector ecology and water quality. Those are two case studies I would like to present how we can apply this framework, the Swiss cheese model, to these type of risks. Let me start with the vector-borne disease example. Here is Aedes albopictus, the Asian tiger mosquito that puts populations at risk for arbovirus infections. It turns out that the climatic suitability for these Aedes mosquitoes has increased dramatically over the years. This is from the Lancet Countdown, the Global Lancet Countdown. This was just published a couple of weeks ago. And here we compare a baseline period from the 50s to the current situations, where we look at an estimated R0 and how that changes over time. And you can see that there is almost a 50% improvement for the mosquito, for the climatic suitability for for transmission for this particular mosquito. Similarly for Aedes aegypti as well. Please don't forget, these mosquitoes are responsible for 4 billion people being at risk for arbovirus infections. This potential disease burden is huge. When you look at last year, we had something like 12 million people that were infected by dengue. So this is a huge issue that needs to be taken into consideration. And there we go. So this is the climate suitability is one of the issues. But work that I had done at ECDC way back, we looked at the underlying drivers of epidemic events in Europe, and we found out that the most important driver for epidemic events, I call them infectious disease threat events, cross-border events, population mobility is the most important driver of these effects. So climate is one issue, but population mobility is the other one. So life is complicated. It's always a multifactorial issue. Here you can see all these people that are flying into Europe. And what is the problem here? You can see that people that go on vacation to Thailand or Brazil, they get bitten by a local mosquito that's infected with one of these arboviruses, Zika, chikungunya, Zika, dengue, and then they fly back to Europe during the summer months and get bitten by a local mosquito, illustrated here on the other side, and that mosquito now is infected with one of these viruses and transmits it to someone else, and this is when we have a local outbreak, autochthonous transmission. That is the problem. And guess what? It's not just the people that are moving around. Our friends are moving too. Where do you want to go? Mountains, forest, beach, or a city? I don't care as long as it's warm. It turns out that mosquitoes don't travel that far, maybe 200 meters in their lifetime. However, they're great at hitchhiking, and we can track their expansion along transportation corridors. And this is a map now that illustrates that incredible expansion of this invasive species that was brought into Italy in 1990 into Genova, and it spread throughout the Italian peninsula over the next decade. Then a decade, 10 years later, it moved into France and Spain and Croatia and other countries, and has been moving ever since. On the bar graph at the bottom here, you can see the districts that have been infested with this particular mosquito, where the mosquito is established. You can see how in 1995, 20 NUTS3 had established Alopictus, and then 10 years later, a slew of other countries had the same mosquito as well. And the bad news with that is that we have also seen an expansion of Aedes albopictus transmitted diseases like dengue or chikungunya. The first graph over there on the left, those are the cases for dengue and chikungunya. And I don't know if anyone has ever seen an exponential curve. Well, if you haven't, this is one. This looks pretty exponential to me. This looks like a terrible increase. And then on the other panel on the left, you can see the NUTS3 regions that have been affected by these type of outbreaks. And again, you see a dramatic increase in the increase of those cases. We then asked the question, how long does it take for the mosquito to establish itself in a NUTS3 region until we have the first outbreak? What's that time span between establishment and the first outbreak? And on the left panel here, or actually, yeah, depending how you sit, you can see that in the 90s, it took something like 25 years between the establishment of the mosquito in a district until we have the first outbreak. But guess what? That time span has collapsed over the years down to 5 years or something like that. And then if you look at the recurrent event, how long does it take for the second event to occur? So in this case, we don't censor the repeated events. And in this analysis here, you can see that in districts that experienced multiple outbreaks, it goes something from 5 years to get the second outbreak to down to less than a year currently. So again, this is a dramatic decline. However, it turns out that these mosquitoes are necessary, but not sufficient for transmission. So what drives this terrible transition? And for that, we set up a multivariate analysis. We set up this time to event analysis where we looked at a number of different variables to see whether they predict these type of outbreaks. And you can see that environmental variables are very much important to predict the transition from establishment till the first outbreak of dengue or chikungunya. And you can see that these urban or semi-urban environments in Europe are 2.5 times more likely compared to rural areas in Europe. You can see that a district that has at least one imported case from Thailand or Brazil of dengue has an 8% increased risk of having a dengue outbreak. Then you can look at health expenditures. A district that spends 100 euros per capita on health expenditures, they have a 20% increased risk for dengue or chikungunya. Hello? This doesn't make any sense. So why do we spend money on healthcare if it increases the risk? That's not good news. We should decrease health expenditures, right? It's probably not that, it's probably even more complicated than that. I don't know the reason for that, but we all assume that it's in fact the improved detection ability, laboratory capacity, and reporting that explains the better health systems that pick up these cases. In fact, the case of dengue in 2010 in Croatia that was missed but picked up by a German traveler who went back home. They couldn't explain that disease, and in fact, he was diagnosed as dengue. And then when they went back to look at the area where that individual had been, they found all these numbers of dengue cases in that area. So anyway, detection is obviously something here. And then the mean temperature, right? So the majority of our cases in Europe happen in the third quarter. So we're looking at the mean temperature. So a 1 degree increase in the mean summer temperature increases the risk for these dengue or chikungunya outbreaks by 55%. Guess what? Mosquitoes are cold-blooded. They don't like it cold. They need to have it warm, right? And so, hey, let me know when it's 18 degrees or higher. They don't like to get around with, when it's cold out. So we now stratified the districts that are colder and those that are warmer. So we take 20 degrees Celsius as a cutoff because that's when mosquitoes are the happiest. And we basically stratified these districts according to warmer districts and colder districts. And you can see that this Kaplan-Meier survival curve now separates very quickly between the warm districts and the cold districts. Outbreak happen much faster in the warm districts that have been infested with this mosquito. And then you can see that those curves separate quite quickly. Incidentally, this is something that we didn't include in the paper. We had a previous one, and the reviewers objected to the fact that these curves had not separated yet because the significance was 0.08. It wasn't quite statistically significant. And we knew, just give us a month, give us 2, give us a year, and you will see those curves separate. Sure enough, we reran it just recently, and those curves have separated. They are statistically significant. Another thing that we looked at, and I think this paper just came out, we looked at the northward trend or expansion of this of Aedes albopictus. And you can see that the yellow curve indicates how rapid this mosquito is moving further north. And it turns out that it's moving at a speed of 60 kilometers per year. there is no end in sight. This guy is just moving so fast. Then if you look at the outbreaks, it turns out that the outbreaks move very quickly. Also, they move even faster. They move at 120 kilometers per year. And we fitted here a quadratic curve onto these relationships. And you can see that the function indicates this trend is accelerating. It's getting faster and faster. What does that mean for population health? If you look at the population at risk, that's now suddenly at risk because they are in the Aedes albopictus risk zone. You can see that we have something like 5 million people per year that are at increased risk. So you see an expansion of that risk towards 5 million people. It's actually 4.9 per year. So it's this huge expansion of the population at risk. Obviously, we did the climate change projections because this is a climate change session. On the left panel here, you see how we projected temperature forward in time. So we don't know what the future holds, right? So we have different socioeconomic developments or emission scenarios, and we combine those into 3 options here: a more extreme emission scenario that we are currently on because emissions are not going down, and then a less extreme one. And you can see how temperature is increasing dramatically in relationship to that extreme, in those extreme emission scenarios. And if you translate that temperature change into the hazard risk, you can see that in those more extreme climate scenarios that we are currently on, we expect something like a 5-fold increased risk of that risk in the coming decades. And so you can see that the risk is going up. So there's more to come. What does that mean for us in public health? We see that that risk is moving further north. What do we need to do? We need to improve our surveillance systems, right? Because we need to make sure that we capture those cases in districts that haven't even been affected yet. So in summary, 1 degree Celsius increase in mean summer temperature, increases the risk. Warming regions are increased risk, and those climate change projections indicate almost 5-fold increased risk, and we're going from sporadic outbreaks to endemic state. Another issue is waterborne diseases. So waterborne diseases is another priority for us because besides the ambient temperature, we also have marine heat waves, as you can see in the Mediterranean there. And what that means, if the oceans warm up, there's more water that evaporates, and it accelerates the hydrologic cycle. And you can see that evaporation leads to condensation, extreme precipitation, runoff, and infiltration. And in fact, that has been documented again in the Lancet Countdown, where we see more and more regions to be affected by these extreme precipitation events, 64% over baseline. And if you look at this framework for IPCC, it turns out that it's actually much more complicated now, because it could be that a little rain event or maybe an extreme rain event can potentially put people that live in the floodplains at risk, because they can potentially be exposed to floodwaters. And it turns out that those floodwaters can potentially drive the rats out of the woodworks that spreads leptospirosis. We can have contamination with coliform. We can have standing water for mosquitoes. So suddenly you have these infectious disease threats that arise on the horizon, and that leads to exposure to vulnerable population and people that are exposed to contaminated water. And so so here on this list here, there are a number of these risk pathways that I illustrate: heavy rain, flooding, drought, temperature rise, sea level rise, and that impacts marine environments where you have extreme precipitation with the runoff. You have algae blooms, cyanobacteria, and fish poisoning, or you have the Vibrio contamination, the oyster farm. So those are One Health issues that are of concern due to these climate extremes. And let me give you another example that we worked on when I was at ECDC probably 10 years ago now. Heavy rain, storm runoff leads to mobilization and transport of pathogens and waterborne outbreaks. And this is the example. So Cryptosporidium is shed by wild animal and livestock, illustrated in those orange bars. And when it rains a lot, those pathogens flush from the pastures into the waterways. And because the parasite, the oocytes of Cryptosporidium survive chlorination. They can infiltrate the treatment system and be distributed in the water distribution system and cause big outbreaks. We looked at something like that in Scandinavia. These blue dots correspond to waterborne outbreaks in Scandinavia. And we set up a matched case control study where we looked at waterborne outbreaks and the preceding climatic conditions before these outbreaks happened. And we found, in fact, a threefold increased risk for waterborne outbreaks if it was preceded by an extreme precipitation event. So what do we do about that? So on this particular diagram, I mapped the climate hazard and exposure and vulnerability on the axis of the cube of the disease burden for waterborne diseases. And we would ideally like to reduce that risk through mitigation and adaptation. Mitigation, again, is reduction of greenhouse gas emissions. In this case, for the water sector, water conservation, energy recovery, renewable energy use, and so on. This is outside of our domain in public health, but the adaptation is within our domain. The WASH system, universal access, wastewater monitoring, and Frank will be speaking about that in more detail, early warning systems that I've worked on when I was at ECDC, and so on, and then adaptation, vulnerability, reduction, by assuring universal health care, climate-proofing the infrastructure, the water infrastructure, so they can sustain these extreme events that the system wasn't built for. So there are things we can do to reduce that risk. So in summary, weather can potentially trigger a sequence of secondary events that are causally connected, and they lead to these cascading risk pathways and cause the waterborne outbreaks. And we need to make sure that our water system are resilient enough to cope with these type of impacts. And this is my final slide. We need to get out of our silos. We need to connect human health with animal health and environmental health in order to make sense of what's going on out there, to come up with those epidemic precursors of disease. So we need to have desilification and bring that One Health concept to different sectors in public health to overcome the obstacles that we are up against. Here's the most important slide. These are the people that have done all the work. Zia Farok and Joachim Rochlöw in Umeå, Kate Lillipot, who is probably in the audience, and Olivier Brier, who's at the ECDC, and then Bernardo Guzman and Karin Nygaard in Norway, who did analysis for the waterborne diseases. And if you have any questions, here is my email. I'm only 1 minute over, I think.
Thank you very much. Thank you very much, Jan. It was a tough message right after breakfast, but I guess you pointed out the reality that we are facing. I think we have still another 4 minutes, so we stick to the script. Tyra, did we get some questions?
Yes, we actually got several questions, and I think there's one that's quite interesting. Now we are talking about surveillance. How can artificial intelligence methods help surveillance of climate-sensitive infectious diseases?
Thanks. It's a great question. In fact, we use artificial intelligence quite a bit for these type of analysis because these machine learning algorithms, they help you to upload a number of different predictors into the model and look at all the different interactions of these variables, which is something that we didn't really do before, right? You remember how tedious it was to code all the interactions of your different variables to see which one is safe and which one isn't. Now the machine does that. And through this Shapley method, you can then rank all those variables that the machine spits out and then see which one is the most important contributor. And those type of models have an incredible predictive power. We have used these models for West Nile and other types of diseases. It's an incredible opportunity, and I wish that we had more capacity in public health of all these IT wizards and geniuses. Unfortunately, they get all hired away by the superpower social media giants that absorb all the talent as opposed for people to work on the common good. So it's a great issue, and I wish we had more capacity in public health to take advantage of that.
Mm-hmm. Yeah, we also have a great answer, and we also have another question, and that's more about this One Health approach. How do we ensure communications and unified approaches across public health and wider disciplines, including, for example, health promotion, incorporating messaging to mitigate climate effects to protect public health? So it's more about how we actually work together to prevent the health consequences of climate change?
Yeah, that's a great question. So it turns out that we have these incredibly sophisticated early warning systems for infectious diseases, for heat waves, and whatever they are. The problem is they don't really work as efficiently as we want. I was involved in the Chicago heat wave in '95, 30 years ago, and we built these heat health action plans to reduce heat-related mortality. Guess what? In Europe, in 2022, 60,000 people died due to the heat. In 23, 47,000 people died due to the heat. And last year, 24, we had more than 60,000 people that died. But despite the fact that we have early warning systems for these kind of climate extremes, they work, but they don't work as well as they should. And so the thing that I've been working on in the past is trying to increase community engagement in this type of early warning system. We need to co-produce and co-develop these type of systems with the stakeholders, with the communities, faith-based organizations, whoever they are, to bring people to the table, because unless people are involved in these kind of early warning systems, they won't buy into the response. So having people early on in the development of these early warning systems and the implementation and the sounding the alarm, people have to be involved throughout the process, and we cannot be complacent. So we need to get public engagement in these kind of early warning systems to reduce the impacts from climate change.
Thank you. Thank you very much as well, Jan. The people here in the audience also have the app, of course, but if someone is not very willing to type and has a very burning question, we could still open for that. Otherwise, before Tyra says her thanks final, one last question, very quick, Jan. Which are the 3 main measures that you would prioritize right now? Very, very telegraphic.
The biggest message is try to connect human data, animal data, environmental data. To me, bridging the disability divide, you might last slide, the silo, coming together and solving the problem of interconnected operationalized data systems. That's what we need. I know it's a pipe dream, but that's what I'm pushing for.
Well, that's the perfect link to the next speaker, because now we will welcome Gerardo Sanchez Martinez from the European Environment Agency. And I know that you're also going to talk a bit about the data that you collect that can be used for this One Health approach. So please, Gerardo, the floor is yours.
Thank you very much. And thanks to all the audience. I'm very, very sorry that I cannot be there with you today. I'm going to talk a little bit about the journey that we are having here at the European Environment Agency in terms of figuring out what One Health means for us, how we can help in this. So a little bit from the institutional standpoint, but also how we use the data to try to advance a little bit. To make the point on what the situation is right now in terms of collaboration across the institution and the sectors, allow me to use 4 exhibits, like in the courtrooms in the American court dramas, right? The first one is going to be the SCIDA registration form. So I had to register, being a speaker and all. And so as you can see there in the field of expertise, you can't really see anything environmental, can you? So I suppose we would be somewhere split between public health sciences, microbiology, preparedness, etc. But there isn't quite a box there for us, right? It's fine. I mean, this is something that happens. This is an infectious disease epidemiology conference, and so in a way, does it make sense? Well, it's unclear, but I mean, I think that tells something about what the situation is right now. The second exhibit is 2 numbers. One of them is the burden of cardiovascular disease due to the environment. So it's about 17% in terms of mortality, not too far off in terms of DALYs, burden of disease. So it's 17% cardiovascular disease. Now, the proportion of environmental submissions in the World Congress of Cardiology in Madrid this very year was 0.00061%. So I think, if I remember correctly, it was actually 2, right, out of like quite very many. So that is the actual representation in a Congress of Cardiology. It is somewhere within prevention. That is the proportion of environmental submissions versus a 17% of the actual burden caused by the environment. The third exhibit is the time it took to sign one specific data sharing agreement between EEA and another EU agency. We were sharing data on chemicals in food. They were very gracious and they worked really hard. They really wanted to help out in this. We were both EU agencies, and you may assume that we share data routinely, but that is very far from the truth, at least currently. It took 17 months with everyone trying real hard, a whole set of lawyers who were really committed to make this happen. It still took 17 months for those files to actually be transferred. That it just happened last week. And the last exhibit is an actual response from a senior EU official to our agency on the need to incorporate environmental risks in a major initiative. And it goes, if you wish to contribute further, you can submit your feedback through the public call of evidence. So just to clarify, anyone can go there. It's called Have Your Say. And like anyone at any corner of the European Union and beyond, anywhere in the world, can put the same submission there than the EEA was invited to put. So that is kind of like the overall current situation, despite all the evidence that we're starting to have, and that Jean went over a tiny bit of it, and it's incredibly complex, but that's the scientific reality. And then we're trying to make this happen in the institutional world, and these are the kind of things we're seeing. So that's the current situation. Now, there's also other labels that we could be using and other approaches. So we could use the global health, so mostly promoted by the United Nations system, the planetary health concept, which has many good sides, many good parts. I think the kind of like the absolute limits element that it has is quite positive. The exposome used, for instance, for exposures leading to cancer. So why is it that we're talking about the One Health approach? Why this one in particular? And I think, it's self-evident that it's because we had a pandemic of a zoonotic origin, and that somehow trickled to the decision makers, so that there was an awareness there that animal health actually matters for human health. That the actors in human health, animal health, and the environment can actually be in the same room, discuss sharing of data, doing joint risk assessments, etc., crucially diminishes the weight of the statement, this goes beyond your mandate. We've heard that 1,000 times. Every time we try to do anything differently, to do things crossover, collaborations that are a little bit less usual or like less traditional, we always say, this goes beyond your mandate. This is not your competency. Why are you doing this? One Health, the umbrella and the awareness, is helping change that a little bit. And that is very, very important in the practice, at least from my point of view, and most colleagues here at the agency share this view. This license to cooperate, so to operate together, is in the practice the most important short-term institutional benefit of the concept. So, for instance, we have an interagency task force on One Health, and the kind of things that we're already discussing and the kind of inroads that we're already making are surprisingly in the best and positive way of the word. We are there with the European Centers for Disease Control. the European Food Safety Agency, the Medicines Agency, ECHA, and it's working quite well. There is really a willingness to collaborate. I think many of us had been there for many years and able to actually collaborate in that open manner. So that is something that is actually improving. Still, because we are at the beginning, we actually get asked quite often, you know, what do you do on One Health, right? As if it were like a specific part of what we do. And this is an incredibly difficult question for us. This is what we're trying to still figure out. The first answer could be, well, all of what we do qualifies as One Health, right? As you know, we work on environmental issues. Most of them have some sort of bearing on human health and on animal health. So in a way, the One Health framework could become a sort of theory of everything on health. It could be one way to go. It's just not very operational per se. The second answer could be that maybe we could start in activities where like the three areas, the three diagrams sort of like converge, right? And then they cross. This is, by the way, taken from WHO. I'm sorry I didn't put the source in there. It's from the WHO framework on the environment in One Health. So if you look at where they intersect, there's clear elements in there. So for instance, what Jan was talking about, the vector-borne diseases with clear environmental influences like climatic suitability or antimicrobial resistance with an environmental component. So those would be undeniably One Health activities. No one, I think, could argue with that. And in that regard, it would be a safe way to go first. But I think my preferred answer is that that's actually the wrong question. Like what qualifies as One Health is a very short-sighted kind of thing. It reminds me of other buzzwords and labels that had their moment of glory and then they died away, right? We don't want that to happen. I think a better question is which ones of the activities could benefit from collaboration under this framework? And the real one is what key human and animal health problems could benefit from including the environment under One Health. I think that one goes to the core of the matter. Now, in that regard, the situation we are at now at the agency, and I know that other institutions are more or less there, is a little bit like what you used to see in medieval maps, right? So you would see like the areas where you know very well the land. So the regular health and environment practice would be those lands by the shore where you know it really, really well. And we know what we're doing and we continue doing so. There's limitations, but we still can do most of what we do. Now, we also can go into areas that we know well are already out at sea, those known areas of application that I was talking about before of environmental elements of antimicrobial resistance, vector-borne diseases, and infectious disease, both in humans and in animals with clear environmental influences. So that we are somewhere between the regular practice and the known areas of application, going a little bit deeper into those areas of application. But beyond that, it's a little bit like in those maps. It's most maps where they didn't know what was there. And so to prevent sailors from going there, they say, here are monsters, right? We just don't know very well what lies beyond, or rather, we have an idea, but we are not clear how to get there and how dangerous that will be. This is an example of a current activity that we're doing. It has very clear and solid guardrails because in the revised Water Framework Directive, there are provisions for antimicrobial resistance. So that implies that both in wastewater and in surface waters, there will have to be attention to these antimicrobial resistant pathogens and genes. And so there you see, we still haven't published this, but we'll be publishing it next year. We are collaborating with a European Topic Center on Health and Environment, as well as the one on biodiversity, which is responsible for water, on looking at the environmental resistome within the One Health continuum. So the part where the resistance happens in healthcare or in food production, households and industry, and there is like a circle there of that resistance circling around. It's well known. There's a lot of attention to it. There's many things that we don't know, of course, but I think there's been attention to that for a very long time. Now, the part where we look at how those pathogens, antimicrobial resistant pathogens and genes live in the environment, are exchanged in the environment, in surface waters, in soil, how they interact with that environmental resistome, and what that means in terms of the risk that might be coming back like a boomerang for both humans and animals. That is something that we still don't understand very well. And so there's some things that we're doing. One of them is, of course, there's been a group led by the IONET network of countries administered by the EEA, where they've been harmonizing some of the methods for sampling this kind of resistance in the environment. And we're also trying to put the message out there to the decision makers that there is such thing as an environmental component of AMR. So that is something that might be obvious for the scientists and the experts, but it's still not in the radar of those making the decisions. And that is something that we're trying to do now. Another area where we are working is on the environmental variables. So Jean has been talking about the climatic suitability and Aedes mosquitoes, but we also are looking into what data we have that could help predict outbreaks of tick-borne disease. And so that is something that we are relying on experts to help us guide our steps and see from what we have at the European level. And we have many, many data streams, including from Copernicus, from satellites, from in situ data. We have data on many types of pollution, industrial and the industrial regulations, air pollution, soil pollution, including topsoil and beyond, surface water, and so on and so forth. We really have a lot of data streams. We're trying to see which ones of those we can use.
My microphone is on. Gerardo, I just need to ask you to start rounding up so that we stick with our times. Apologies.
Sure, no worries. So we're trying to put those data at the service of the One Health activities and concepts and see whether they can help. So here you can see another one of the analysis that we've been going through. You can see in the European Climate Risk Assessment and the health cluster, the prioritization in terms of urgency to act and risk severity there, where regardless of the risk severity assessed in there, just wanted to mention that in comparison with other areas, say for instance the pathogens in coastal waters, the attention to infectious diseases per se is actually much more clear in the plans that have been analyzed. One of the ways we're trying to at least send the message that there needs to be an integration of environment and health data is the European Environment and Health Atlas. What you see there is one of our most popular widgets, which is the Check Your Place, where you can insert your workplace or your home address and see what the air pollution is there, major pollutants, you can see what the noise is like, what the bathing water quality is nearby, and what blue and green spaces are there. It's proven quite popular with subnational authorities, so please have a look. That's the QR code. And in short, we know some of the ways in which the environment can be incorporated into the One Health framework, but we need to figure out whether it's going to be in the practice, the theory of everything, a license to cooperate, whether we focus on infectious or also on non-communicable diseases. We do see hopeful signs of cooperation, and so we need to continue working on those, particularly things that are working, so that they become successful examples of things that can happen if we apply this process more fully. Thank you very much.
Thank you very much, Gerardo, from this interesting talk. I think we'll have to move to the next speaker.
Yes, apologies. We want to stick to time, but Gerardo, I want to say, and I'm looking at my fellow chief scientist from ECDC, so I don't want to put him on the hot spot, but I think next year we will find an environmental box to click when you register for Escaide. So you will be more motivated even to join us and discuss more environment here at the conference. Thank you very much. We change a little topics. We stay online with Denmark, and in this week where we talk of AMR, we could not avoid going a little deeper into AMR, and for that we have Professor Frank Aerstrup from the Danish Technical University, who will indeed for the next also around 10-15 minutes take us on a deep dive about this challenge also to the environment. Frank, the floor is yours. But we cannot hear you yet.
I'm muted. Okay, well then it's going to be a very, very, very quiet talk. But anyway, thank you, and sorry for not being there in person. We have a research evaluation today, and I have to be present there. And also, this is going to be relatively short, and it's going to be a bit, egocentric. And I apologize for that because there is a lot of work going on around the world, and of course, this is not only something we are doing. But I think that we cannot underestimate the potential of actually sewage in being a part of the surveillance for the future. I think if you look at it from a perspective of what is it actually we need, well, it is surveillance. And I think that the two previous speakers have also led to this, that in order to know where we are and where we're going, we really need data. And that has to be geographic, reservoir, and pathogen independent. We cannot predict what is going to be the next problem. So if we just focus on these few notifiable diseases or that single reservoir, we are very, very, very likely going to miss what is actually going to happen. And I see a lot of studies right now where people try to rewrite history, focusing on a few, top pathogens, but that's not really helping us in terms of actually preparing for the future. It's fine to learn from the history, but we really need to know what's happening also for those that are novel emerging pathogens. So very rapidly observe trends and transfer that information around. And we definitely do believe that sewage can play a role here. So of course, where should we get the data from? And I think that very often today, where we get our data, that is really at the top of the pyramid. That is excluding the healthy population. This is very much looking at hospitalized patients, and often also when it comes to antimicrobial resistance, where people have already been exposed to antibiotics. So it's very, very likely also a little bit overestimating, what is actually the problems and biasing this towards some of these carbapenems or other multidrug resistant bacteria. So it could be a wonderful way of actually bypassing this problem. There's also a lot of ethical concerns there. In Denmark, we have for a very long time tried to go out and see if we could actually do surveillance of healthy humans. It's basically impossible. Getting healthy people to bend over and deliver a swab, that is very difficult. But sewage is something that we have been looking at. I think that when we start discussing this from our side, it's a little bit more than 10 years ago, where we could actually say that as soon as people deliver a sample here, it's not theirs anymore, it's considered environmental, and we can do whatever we want to do with this. And the first thing we did was actually to look into long-distance airplanes, but this is more than 10 years ago. We do use and do prefer to use a technology called metagenomics along these lines with not missing anything. It's where we go out and completely randomly sequence everything that is in a sample. That, of course, also means that we have a lot of noise and we need to do a lot of analysis afterwards. I think that from what I've seen at least, then there's also a very, very, very common mistake people do when they're looking at metagenomic data. That is that they just consider the resistome as one single entity. So they look at what are reads mapping to a specific database, and the more genes, well, the better. At least that's what they assume. The problem is that a lot of the databases we have out there, they include a lot of intrinsic genes that are just species markers. So from a personal perspective, I think that perhaps 50 to 80% of all the publications you're looking at are flawed because they are simply mapping to genes that are species markers. So when they make conclusions on the resistome, it might just as well be the species composition. It is a bias, it is a problem, it is something that of course needs and can be addressed, but it's just something that really you need to be aware of if you're just looking at the resistome as a random number. As I said, our first study was published in 2015, and don't go back to that because it has the same bias as I'm criticizing now. But nonetheless, at least for us, it showed that this was probably feasible in terms of actually using sewage as something for global surveillance. So the year after, we decided to go out and see if we could collect sewage from around the world. And this is what we call the Global Sewage Surveillance Consortium. It's been running for quite a while, from 2016 to 22. Data from 350 cities in 111 countries. And just briefly, in terms of what have we experienced during this, and of course we also published, but I think that there are definitely a higher abundance of resistant genes in some countries, and as seen on this, it is very much around the tropics, but it's not only around the tropics. There's also other countries. So this is not only associated to temperature, that there are definitely other drivers around this. We do see, surprisingly to us, still a regional separation in the diversity. We're not talking about abundances here, but which genes are prevalent, even if they're giving resistance to the same classes, then which genes are prevalent in the different countries. So there's definitely some regional separation here that cannot just be explained by usage or hygiene or something else. There is a regional signal here. There is also a huge reservoir of latent antimicrobial resistant genes. This is genes that are still, as far as we know, located on the chromosome, but if mobilized, can then cause resistance. And that diversity is everywhere. There's more, strangely enough, actually more latent resistant genes in the tropics, but nonetheless, you can go to Greenland, you can go to Africa, you can go to basically probably Antarctic as well, and you will find genes there that can cause resistance if they are mobilized. So there's a lot of reservoirs out there that we, even to antibiotics that have not even come into the clinics yet. And then also, if we look into the details, and I think you'll have to trust me here, but if you look at each individual resistant gene, they actually behave differently. So we cannot say that there is a epidemiology or evolutionary trajectory for the resistome. It is per antimicrobial resistant gene. Some of them are globally dispersed, some are not, some are located on some mobile elements and within certain regions, etc., etc., etc. And it all differs according to the different resistant genes. And then also we do see some of these strange ecological effects, where we call it distant decays, where there's variations around the world. So in contrast to what I have believed some time ago, that everything is everywhere because we have this rapid transmission around the world, there does seem to be a little bit slower transmission also of resistant genes, and actually picking up mutations as they move around the world, and just looking at the geographic locations there. And then, at least in our hands, we can see that AMR abundances are better described by socioeconomic factors than by antimicrobial usage data. A big question is, of course, how does sewage actually relate to clinical AMR data? That has been addressed in a few studies, surprisingly few, actually, and I think this is also something where we need more data. This is unpublished data from our hands, where we went out and collected clinical data from 33 countries, so simply went out and got isolates and got them to Denmark and sequenced them, and then compared to our sewage data from the same countries. What we can see is, if we look at the antimicrobial resistant genes, well, it's not all antimicrobial resistance genes that are present in both, collections. Some are found only in clinical isolates, some are found only in sewage, but of course there is also an overlap. Then also there is what you call a non-random effect. There is a lot of noise here. But if we assume that the distribution of genes were just random, it's not. There is a correlation if you look at the country level in terms of which resistant genes are found in clinical isolates and which do we see in the sewage. And there is also a good correlation of gastrointestinal pathogens and for some antimicrobial resistant genes. But for others, there's absolutely no correlation. You could even find negative correlations. For some genes, it's good. It's overall, it's there, but with a lot of noise. But I really think that we need to look at this as a per gene associations and not just talk about resistome per se. And we have also verified this in other studies when looking at epidemiological data. So concluding remarks, surveillance is the base of everything, and it is extremely important that we get a better global surveillance. Sewage surveillance could be a very valuable contribution, but only a contribution, not for all pathogens or all resistant genes. But I think it does have this wonderful thing that you can go back and reanalyze it, like looking for, oh, here's a new gene. Was it there? Where is it? And so you can get a lot more information out of sewage. There are very critical decisions to be taken regarding methodologies. Do you want to do single isolates, selected genes, for example, qPCR or metagenomics? That is definitely my favorites, but also the most expensive one and the most technical demanding one. And there is a lot of decisions around correct sampling, DNA purification, and the bioinformatics. And unfortunately, as far as I can see, most of the studies published right now simply ignore the part around, especially the databases they map to. And then perhaps a personal remark, I think that the step from research to implementation has happened a bit fast and without sufficient knowledge transfer. I think that it's wonderful to see, of course, the political decisions that now you need to do AMR surveillance in all countries across Europe. I just think that there is some caveats there that people are perhaps not aware of. I also feel that perhaps a little bit more involvement of some of the research groups around Europe would have been good, for example, in this EU WIS consortium and others. So I think that perhaps it has happened a little bit too fast. It's wonderful, it's good, it has potential, but it also needs to be implemented in the right way. So thank you.
Thank you. Thank you very much. Thank you so much, Frank. Right on time. Thyro, we may have time for one or two questions.
There are no questions in the app, but I have a question for you, Frank. So you mentioned that maybe we do lack some knowledge before implementation that has gone too fast. What do you consider as the three main knowledge gaps that we have when it comes to wastewater surveillance?
Yeah, I think that the correct way of analyzing the data, you might say. I think that basically all studies I'm looking at right now, and that is in Nature and Nature Communication and Nature Microbiology, they simply ignore that when you look at the resistome, then some of the genes are just species markers. And even if you as a reviewer, criticize this, then it actually gets published anyway, which is a bit of a frustration from some and I cannot review everything. I think it's a big frustration, and it's simply it's moved a little bit too fast, and people publish without knowing what they're publishing on, unfortunately. And it's not only in the surveillance area. It is definitely also in other clinical studies. So I think that understanding what is actually, defining what is a resistome, which would be very great. Then I also think that what is the role of the environmental surveillance? So what does it actually predict? So what's the value? I think that we could have gone a little bit, further in terms of also understanding this. And then I also think that what we call, the governance around the data, who is collecting them, for what purpose, and how can they be reanalyzed and reused? That is also something that is missing. And I've seen it before. It was the same when you implemented, integrated surveillance back in the mid-90s. It was also like, okay, suddenly everybody's doing it. It was needed, but also there, I think that perhaps it happened a bit too fast. Yeah. So and sorry for being such an old conservative, whatever, but I guess that comes with age.
Thank you so much for this clarification.
We have a question from the floor.
We have one question from the floor.
Thank you, Frank. I'm Maarten de Jong from the Public Health Service Amsterdam, the Netherlands. I think we can all agree that wastewater surveillance is the future for infectious disease control and management. I'm not biased, maybe a bit, but what I miss in the One Health approach is that the potential that wastewater surveillance has. And I wonder what is needed for wastewater surveillance to become a key priority in this approach. And a second question is, you mentioned in your last slide that wastewater surveillance is not feasible for all pathogens. Well, we don't know yet. There's so much potential that can be explored. So those two small questions for you. Thank you.
Yeah. Well, I think that you're absolutely right. Well, perhaps it's going too far to say that it's not feasible for everything. It could be, definitely, there could be. But I do think that at the current state, at least, the sensitivity is insufficient for a lot of the pathogens, and also the correlation, you might say, to clinical data is definitely lacking a bit there. So I think there's more work on the sensitivity, the low abundance reads, and what are their correlation, the precision, because when you do bioinformatic mapping, you're also getting a lot of biases. I think that's needed. And what was the second question?
He was also asking about the integration. How can the sewage surveillance become more mainstream in One Health as well?
Yeah. So, but I think that's perhaps where we probably are somehow lacking. What do you call it? I don't know if you call it an European, zoonosis center or some coordinating body. I do think there is a need for an office. It could be almost like a single person that is actually trying to coordinate this. I think that Gerard, also mentioned this, that, this is not within your mandate. But now we need to have someone that actually has the mandate to do everything and to coordinate everything. So I think we need a coordinating body for Europe. I think that we need to, it's artificial to have, public health here, animal health here, food safety here, and then environmental here. That's not, well, this is human artificial bodies we have made that does not reflect nature, and we need to have someone that can actually coordinate this. A lot of countries have, we have in Denmark the Danish Zoonosis Center that has been trying to coordinate integrated surveillance with some success at least. Other countries have similar things. But I think that we need a coordinator for Europe now.
Thanks.
Yeah, so this underlines the importance of the One Health.
And I will not volunteer.
Thanks a lot, Frank. I think we have to round out and move to our final speaker. And I'd like to welcome Bodwin Katri from Semenza, who's going to talk about the One Health AMR surveillance program, BELMAP.
Thank you. Thanks for the organization for having me here in this nice venue. I like to talk about BELMAP, and BELMAP is inspired by the previous speaker, Professor Aarstrup, who was among the people that made DANMAP in Denmark, one of the pioneer One Health items in this subject. By reasons I do not completely understand, Moira Kelly, who is here indicated with the 2 stars, assembled all these people in Belgium in a language which is not an official one in our country, the English language that I myself do not yet master. And she succeeded to have all these stakeholders on board, people from food, people from human health, people from veterinary health, and people from environmental health. So one question I would like to answer with you is by whom and where are most antimicrobials consumed and thereby selecting for resistance? Because this is one of the main objectives of this report. Here you see two of these sectors. In blue, the human medicine. In green, the animal husbandry. Remind, it's livestock. So you see a huge decrease since 2012 in livestock, and human medicine is quietly stable with one exception during the pandemic, a drop which has been seen in many countries. This is the absolute volume used. And of course, and for wastewater is the same thing, you need to have a good denominator. So to what do you compare this volume? And therefore, we use kilogram biomass. And if you take the same information, but you have in the denominator the biomass, you can see the graph below. And here you see actually that far more antimicrobials are given to humans when you adjust for their body weight compared to animals. You also see the trends. If we now go into animal, so please remind that there's a huge decrease in both graphs, in green, in the animal husbandry sector. So if we now go into that animal sector, we see here for 4 categories of species, beef cattle, pigs, poultry, and veal calves. In red, the things that we do not want, multi-drug resistance. So if it's declining, it is good. And it is declining. It is in line with the decrease in consumption. We see in green what we want, pan-susceptible E. coli indicator bacteria. And you also remark that we have, for instance, from ruminants, beef cattle, and veal calves. But the veal calves, as you see, far more multi-drug resistance compared to beef cattle. They are not as innocent as they look. And we remind also that dairy cattle, so the cattle that produce milk, are not included. And there's a simple reason for that. That's because you cannot give antimicrobials to a cow because otherwise the milk and the fermentation products will not be possible. So 1 drop of penicillin would destroy an entire milk tank. So we do not measure them as detailed as the other animals. And I also think that is a point of One Health monitoring. Do not measure what is not needed or avoid unnecessary sampling. Let's get back to this slide. The denominator is very important, not only to compare human and animals, but also in the environment. But let's focus now on human medicine and only focus in the light blue evolution of hospital antimicrobial consumption. And if you take a good denominator, a little bit tricky, the number of patient days, then you see even an increase in Belgium. On the right side, you can, for your information, see that the universal hospitals, here called the tertiary hospitals, have the highest amount of antimicrobials, but they're doing better the last few years. So you can have, in that Belmont report, reference to the detailed reports too. The targets for Belgium on AMR have been established by the European Union, and we do not meet them. Here, 3 bugs, E. coli, Staphylococcus aureus, and Klebsiella pneumonia. We have been there during the pandemic, but now we are in a rebound phase, and in many other countries it's the same. So we are not there yet. We see large increases in sexually transmitted infections, but there is a session later this day. What I hope, my dream, is that we have a geographical information system, a geo-mapping system in which all this information, similar to the previous speaker from the European Environmental Agency has shown, that you can just have the information by simply clicking where you are. And that should be, in my opinion, not restricted to the nomenclature and the visualizations that microbiology has. It should be something that everybody can read, even the children, even elderly, and so on, because they can help us in these matters. They can report. Some of them did during COVID. I think this patient empowerment, this society empowerment is important. We also have, since a couple of years, environmental data in our Belmont report, but it is still ongoing. We have retrospective data of the azoles, antifungal, and the majority is used in agriculture. You see that there is also a rebound after the pandemic. And at your right side, 2 huge increases. So in 2022, you see a lot of human hygiene disinfectants were used, biocidal sales. And then in 2023, the last available data. So we argue for more recent data. And that is something I think the European Commission should take care, that we reduce the administrative burden, as we have also seen in the previous speaker, so that researchers can again do research instead of having all these documents, templates filled out. You see here that in dark green, surface disinfectants are incredibly increased. Again, veterinary medicine is increasing its use for biocidal. And from a point of monitoring, it's good that we have that information, but as a Darwinian believer of the selection of evolution, I think it is only a matter of time before we see huge amounts of biocidal resistance in the target pathogens that we want to conquer. So that is a concern. And the next concern is sustainability. At your left, you see all the disposable equipment which is needed for a sample to confirm that somebody has a bloodstream infection. At your right, you see material that is needed in the lab. We have now in Belgium sustainable coordinators in the hospitals that want to decrease these amounts of disposable things. I think in the next generation, this might also be applicable for the microbiology lab and for all our jobs that we're doing. And I would like to end with saying that Belmop is available for you. It is your one-stop shop for antimicrobial consumption resistance, trends, recommendations, and it is interactive. So you can scroll and find the information. The new report will be launched tomorrow. So look today and again tomorrow. And I thank Moira Kelly and many other people involved and you for your attention. Thank you.
Thank you, Bodwin, for this impressive system that you have built up over the years. Are there any questions?
Let's see if people start to type. But of course, I had one. I've seen the slide you added on the environmental components, namely on the fungicides and biocides. But I'm wondering, Bodwin, I mean, in the Belgium context, have you found a better solution to do this integration? Are these ad hoc surveillances? Do you have it structured in the system that you follow up resistance for these traits as well?
No, not yet. All these compiling is information available, but there is not yet a law that integrates these. So this is really voluntary work from people that have a mandate in the business. Mm-hmm.
Yeah, because that links, I think, very much also to a question, the last question here from the floor, on how to bring sewage. And I think here the power of regulation and the power to legislate. And I'm putting my European Union hat just to think about the water directive, for example, with the requirement now to surveil for AMR. We need sometimes enforcement, because it is quite, well, not scary, but concerning elements you show there. I turn also to the room to see if there are any further questions. Otherwise, we still have a final minutes here to do a little bit of interaction with all of you, the audience, and still a few more questions to our colleagues. So yeah. Thank you very much again, Bodwin.
Thank you, Bodwin. So now we'll move to a Slido poll. So I hope we'll get it on the screen soon. Yeah, you can see it here. At least we can see. Yeah, there's the QR code. So we have some questions that we would like to ask you. And the first question is, in your country, is there collaboration in place with the environmental sector related to surveillance, research, or risk assessment? And there are three options. Yes, there's a formal collaboration. Yes, there's an ad hoc collaboration. Or the third option, no, there's no collaboration. So you'll just have a minute to answer the questions. And we're so excited about this.
Let's see. Who wins? See, 90 have answered. The number is going up. Let's go, people.
Yeah.
There's too many of us in this room. We need to break records. Yeah, quick.
Still people coming in. Mm-hmm.
We'll give them a little more time. I want to reach 200.
Aren't we excited about this? I think maybe it's time
let's see the results.
Get the results. Okay, so what we can see here is that almost 1/3 of the answers, there is a formal collaboration, but mostly it's an ad hoc collaboration, and it's actually quite encouraging, I think, that in only 20% of those who have answered said that there's no collaboration at all. So I think that's quite encouraging to me. So.
Yep. And we can open here also briefly to our guests if there's any feedback from the two here or those online, both Frank and Gerardo as well, in terms of this collaboration. Perhaps to comments, how do you envision ad hoc? How do we get the ad hoc to permanent?
Well, in Belgium we have a 5-year national action plan on antimicrobial resistance. A new one is ready to be launched, and in there the BELMAP is included. So it is an ad hoc which is included in a national action plan.
Any more comments? I don't see Frank and Gerardo, but if you have some.
Well, this is Frank. Well, we used to have a relatively strong collaboration in Denmark, and I think that has somehow diminished, over time. But back in the early 90s, we implemented a coordinating body as the Danish Zoonosis Center, and I think that actually worked very, very, very well for a number of decades. There is still some legacy collaboration coming out of this, so it's not like it's totally disappeared. But I think that we have not really done a good job of keeping it functional and expanding. So I think that's I think that we have rested a little bit on our laurels, you might say, in Denmark. We still collaborate, but it's more on this is what we used to doing basis, unfortunately, and especially the engagement, of research, public health, veterinary health, environmental health. That is definitely greatly lacking in Denmark. Something we should do something about.
I think we have a formal collaboration among the organizations in Denmark for environment, animal, and human health, but at the top level. Gerardo, would you like to comment?
Yeah, I would say there is wisdom in the crowd, as in ad hoc, I think describes accurately the current situation also at the European Union, but there is serious efforts to graduate beyond that. And I think some of those are already panning out. I mean, I'm thinking about joint risk assessments, like for instance, of the antifungicides that happened recently. And so in that regard, I think there is serious efforts to go beyond, but the current situation can accurately be described as ad hoc collaborations.
Thanks, Gerardo. I think this ad hoc is also sometimes important for the low-hanging fruits. And of course, I may sound biased as an European agency, but the example of our 5 agencies coming together, trying to transform ad hoc into something more permanent, is a good example. I won't speak about it. I will perhaps very bluntly say there is a fireside in the afternoon. So if you want to know more, you should go there. But we do have one last slide before you're all given a chance to boost on caffeine. And this second slide, which we will now put on the screen, takes us to one thing that has been mentioned here, I think, more times than any. Only environment word has been repeated more than the word data. And for that, as soon as the question comes up on Slido, perhaps it's already there, it's really about, in your country, do you use or collect data from the environment for the purposes of surveillance, research, or risk assessment? And let's see if ad hoc still wins this time.
So Bodil, I suppose you would say that you do it on a regular basis, right? Even though it may be once a year. I don't know how often you collect the data.
It's yearly collected. But we aim to have a real-time system, but for the moment, that's still in our development.
Okay, we'll have another 20 seconds before we need 25 seconds before we need to close the poll. So let's have a look at the result here as well, please. And the winner is...
Ta-da!
Wow. Now, anyone surprised, speakers? This would be another session of discussion now, I think. Because with so many collecting it regularly, it raises the question, why are we not doing more with it? I think that was also highlighted.
Yeah, it's also interesting that we have more people collecting data regularly than having a formal collaboration or having a regular collaboration. I think that's interesting.
I think we should have a follow-up question to see how many people actually use the data, because collecting data is a good thing, but the Achilles heel of surveillance, if you don't use it, that's the worst, right? So do people actually use it for their analysis? That would be a better question.
Indeed. I think we are reaching the end of our time, Tyra.
Yeah, so coffee is around the corner, but I would like to thank all the speakers for your participation and you contributing to this important discussion.
And from my side, I want to thank, of course, everyone that joined this morning for this on environment. We talked a lot about how we bring environment more to health, but there was actually a question which we didn't manage to answer today here because there were too many. Thanks to everyone for the many questions. How do we also bring more health, whether it is human, animal, to the environmental sciences? How can we also populate their congresses? Let's keep that in mind. And again, thank you very much for joining this session. And I give the floor back to our Master of Ceremonies, Martin.